Alright, my prediction for this lab is that oxygen levels will steadily increase as time goes on and the plant produces more oxygen and consumes more CO2 in the water. Here's a graph of the oxygen level versus time graph, and I predict it will steadily increase before leveling off when the water becomes saturated with oxygen.
Photosynthesis consists of two cycles. Number one would be the light cycle, number two would be the dark cycle. In the light cycle, chlorophyll molecules store energy through the sun into the chloroplast, which creates ATP molecules. These ATP molecules release their energy and form the water and CO2 molecules that enter the plant through the stomata. Based on our observations, we have found that our hypothesis is completely wrong. It is actually the exact opposite of what we intended. Now, we saw that right when we put in the plant, the oxygen levels immediately started to be below when the water was by itself. And at 50 seconds, which is right about there, we put the light in, and we put the light on the plant, and the oxygen levels even started to decrease even more, which was completely what we did not expect. And after...
Alright, so the results we got here were not what we predicted our hypothesis would be, but we think we may have found out the reason why. From looking at another group's data who had a temperature probe in it, the temperature steadily increased as the light was put in there, and we did a little research and we found that oxygen dissolves less in warmer water. So as the water was getting warmer, the amount of oxygen that could dissolve in the water was decreased, so that offset the amount of oxygen put in by photosynthesis. Yeah, that's what we think happened in our graph. So to make the experiment better, it probably would have been better to make the water a constant temperature so the water wouldn't heat up anymore from the light. And
in order for this hypothesis to be correct, our water temperatures must have been constant, and that would have allowed the oxygen levels to rise. But since the water was heated, it just can't happen. It's not plausible.
So basically, some of the things we can learn from this experiment are that the results aren't always what you think they are, and that you need to take into account all the variables, even ones that you wouldn't think, like the temperature of the water.
Our predictions for the lab is that the graph will show a decrease and an increase from the results.
Just like at first with no plant inside it, with like nothing in it, the photosynthesis is slowly going up, and now it started to go down again. It started to go down when we put the plant inside. So we think it's going to go down for a while, and just shoot back up after a little while.
So after 15 minutes of run number 2, the dissolved oxygen levels have dropped dramatically, as you can see here. Represented in the green line, run number 2, from run number 1, the red line.
You are the background of experiment.
The dissolved oxygen level was on a steady decrease, and that's about it.
Better, okay.
So, contrary to our hypothesis, the results show a major decline in dissolved oxygen levels, represented by the green line. And from the results, we inferred that because with the light on, carbon dioxide levels increased and sped up the reaction to CO2 in our plant, and therefore caused a decrease in dissolved oxygen in our results.